Okay, here we are back for the 7th lecture here on chapter 21. We've come quite a ways and hopefully you're enjoying all of it and you're ready to move forward into nucleic acids and protein synthesis. Alright, so our particular focus here for the 7th lecture is protein synthesis, and in particular it's translation. So if you're a foreign language scholar, then you have a pretty decent understanding of translation in that realm. So let's see how you understand translation as it relates to protein synthesis. Alright, so the transfer RNA activation occurs when a synthetase uses energy from ATP hydrolysis to attach an amino acid to a specific transfer RNA, tRNA. It prepares each tRNA to use a triplet called an anticodon to complement a codon on messenger RNA. So we had that term anticodon before and we didn't go into it because it wasn't time for it, but now we're going to see a little bit more about the anticodon. If we look at the 5' to 3' codon on the messenger RNA, we end up at that bend there at the bottom of the molecule as it appears on the screen with the complement. The codon on the mRNA gives the anticodon. So UCA complement would be AGU, the anticodon on the tRNA. Now, if we talk about initiation and chain elongation, the initiation of protein synthesis involves an mRNA binding to a ribosome. The start codon, AUG, binds to a tRNA with methionine. Previously, our tRNA there had serine attached, but this time we're looking at a tRNA with a methionine. The second codon attaches to a tRNA with the next amino acid, and a peptide bond forms between those adjacent amino acids at the first and second codons. So we're starting to make our polypeptide. Our next step is translocation, and during translocation, that first transfer RNA detaches from the ribosome. The ribosome shifts to the adjacent codon on the messenger RNA. We get a new transfer RNA with amino acid attached to the open binding site. A peptide bond forms, and that empty tRNA now detaches, and the ribosome moves down the messenger RNA to read the next codon, sort of like a protein assembly factory. And so here we see a cartoon of that process, the whole thing, right, from the DNA unraveling to the mRNA binding to it, the mRNA leaving the nucleus, attaching to that ribosome, getting translocation to start. We've got our growing peptide chain there coming in, the incoming transfer RNA hydrogen bonds to the complementary mRNA sequence. We get the codon anticodon thing going on that we already discussed, and we add these amino acids one by one to that peptide. The tRNA brings them in, and they're combined, chemically bound there through the ribosome in the direction of ribosome advance. Now everything comes to an end, so the termination of protein synthesis happens when we get a polypeptide with all the amino acids for a protein synthesized. We're ready to stop, so the ribosome reaches a stop codon, either UGA, UAA, or UAG. There is no more transfer RNA with an anticodon for the stop codon, so that's how the polypeptide releases from the ribosome. There's nothing else left to bring another amino acid through a transfer RNA. So the polypeptide releases, the ribosome is ready to do more work. Okay, so it's a relatively short lecture, but a lot of big concepts here. So let's have a couple learning checks. In this first learning check, you're going to match the following terms to their definition. So we have 1, 2, 3, 4 for the terms, and then A, B, C, D for the definitions. Go ahead, stop the video while you link A, B, C, D with 1, 2, 3, 4, and then start us back up when you're ready to check your work. Good luck. Okay, and hopefully you arrived at the same answers I did here. So A, ribosomes move along messenger RNA, adding amino acids to a growing polypeptide chain. That is 3, translocation. B, a completed peptide chain is released. That's 4, termination. So the release should clue you in that we're at the end, and that's what termination means. For C, a tRNA attaches to its specific amino acid. That's one, activation. That's the tRNA getting ready to do its job by bringing that specific amino acid in. And then D, a tRNA binds to the AUG codon of the mRNA on the ribosome. That AUG codon, very important because it is the initiation codon too. So it's telling everything to get started. So there we have it. Hopefully that's what you had in your selections as well. If not, hopefully you understand now why those are the best choices. And if that isn't the case, then hopefully you'll reach out to me for some help. All right, before our final learning check for this lecture, let's summarize protein synthesis. So first, an mRNA attaches to a ribosome. The molecules of tRNA bonded to those specific amino acids attach the codons of the mRNA. The peptide bonds form between the amino acid and the peptide chain. Our ribosome shifts to each codon on the mRNA until it reaches that stop codon. And then finally, the polypeptide chain detaches after termination to function as an active protein. So that's the summary of that overall process. Okay, so now that we've looked at protein synthesis in some detail, let's end with a final learning check. And here we're asked the following sequence of a section of DNA is used to build an mRNA for a protein 3' GAA CCGC TTT 5'. So what is the corresponding mRNA sequence? What are the anticodons for the tRNAs? And then what is the amino acid order in the peptide? So obviously you can go back and look at that table from our previous lecture because I wouldn't expect you to remember all of them. Otherwise, I'd give you a small number to choose from as I did in the previous learning check and at the end of last lecture. So don't worry about memorizing all of them, but please do consult that table to answer C. So I think it's going to require you to stop the video here. It may take you a little while before you're ready to start back up and check your work. Good luck. All right, so there for A, hopefully you identified, of course, if we're starting 3' to 5', then our corresponding mRNA sequence is going to be 5' to 3', and it's going to be CUU, right? We're RNA, so we don't have any T's. Our CCC is GGG, and our TTT is AAA. So that's our messenger RNA. The anticodons for those tRNAs would be those three sections, right? The mRNA codons are what we just described above, and therefore the tRNA anticodons would be wherever we have a C, we go to a G, wherever we have a U, we go to an A G goes to C, and an A goes to U. So now, what is the amino acid order in the peptide? Well, we look at those messenger RNAs and the codons, and we see that we're going to end up with the peptide of leucyl glycinyl lysine, LEUGLYLYS. So hopefully that was, it may have taken a little while, but hopefully it was relatively straightforward, especially since you don't have to memorize all those mRNA codons. So if you're struggling, you're just frustrated and not sure what's going on, I was there, trust me. Biochem is not my forte, but I certainly enjoy it, and I've come to like it and understand it a lot better now. But if you struggle with it, know that I struggled first, and I'm happy to try to help if you reach out for some help. If you're a biological chemistry whiz and everything's going very well, that's great, and we'll meet again in the next lecture.